We're live now. Welcome to the concluding session of 4th edition of Health Tech, presented to you by Bengal Chamber of Commerce and Industry and Medical Super Specialty Hospital. Our knowledge partner is PwC, and this program is being presented in association with Woodlands Hospital. This session is Technology Adoption in Healthcare Delivery in Immunology and Rheumatology. So I would request the chairperson of the Health Committee of the Bengal Chamber, who is also Senior Vice Chairman, Cardiology Services and Senior Consultant, Interventional Cardiologist, Medical Super Specialty Hospital, Dr. Robin Chakraborty, to please introduce the other speakers and take the session forward, along with the chairperson with this session, Professor Dr. Kanjaksha Ghosh, former Director, National Institute of Immunohematology, ICMR.
Thank you, Angana. Thank you. So as we all know, this is the last session of Health Tech 2021, 4th edition of Health Tech. This is a meeting which is going on for 4 years along with the Medica Super Specialty Hospital and other hospitals of the city. So, but it is a forum where we actually talk more about technology rather than the clinical thing. Mostly made for the common people, other professionals. So, and the previous session was really great. People talked on cardiovascular technology, and the session before was also an excellent session on the technology application, especially the digital technology in healthcare. That was great, especially the talk from New York was excellent. So in this particular session, the session will be chaired by Dr. Kanjaksha Ghosh, Professor Kanjaksha Ghosh. He's a phenomenal person from the knowledge point of view, from the medical knowledge and its application and the technology, particularly in relation to immunology and hematology. He is now former director of National Institute of Immunology and Hematology, ICMR, Mumbai. So may I request Professor Kanjaksha Ghosh to start the session and introduce our speakers, Dr. Amit Dinda, Dr. Shiva Prasad Rai Chaudhary, and Sritikana Rai Chaudhary. Dr. Kanjaksha Ghosh, chairperson of this particular session. Professor Ghosh, please.
Thank you, Dr. Robin. And actually, what is the order in which Dr. Amit will be speaking first? Hello.
Shiva Prasad Rai Chaudhary is speaking first, and then Dr. Amit Dinda, and then Dr. Sritikana Rai Chaudhary.
So the introduction, I think you do, because I do not have the details of each of them. Then I will be talking.
Perfect. Thank you, Dr. Ghosh. Shiva Prasad Rai Chaudhary is a professor of medicine immunology in California University of Davis, but he has a background of his basic training from Kolkata Medical College MBBS, and then MD from PGI Ahmed Chandigarh. Then he was in USA and has developed himself in the area of immunology and immunology and the clinical medicine. It is a great honor for us to have Dr. Rai Chaudhary here in this particular session. And
yes, Professor Rai. Yeah. Professor Rai Chaudhary, please.
Thank you for giving us the opportunity to talk in this forum. You can see my slide?
Yeah, it's all right.
Okay. So it's a great honor to be here. And, I was just talking among ourselves.
Go to the slideshow. So we don't need to see the other slides. Click on that. That's it. Okay.
So we are just talking about ourselves that Dr. Robin Chakraborty, Ravinda, put us all together. We all grew up in Kolkata, and then we moved to All-India Institute and PGI, and some of us moved to both of the places. And now we are meeting, this group is meeting after 30 years. So we were young and very energetic on those days. So I'm remembering our fun days in those times. Thank you again. So I'm going to talk, it's a technology forum, as mentioned by Professor Chakraborty. So my title of my talk is Molecular Imaging, a Diagnostic Tool for autoimmune diseases. So how do we define autoimmune disease? Autoimmunity is an immune response directed against an antigen within the body of the host. The definition does not distinguish whether the response is innate or acquired, and if acquired, whether it is induced by a foreign or by an existing in vivo antigen. These are heterogeneous group of inflammatory, generally chronic disorders of unknown cause. This affects multiple tissue and organ system. So in many conditions, the whole body gets affected and it becomes clinically challenging to take care of these conditions. The basic principle, immunological principle, is these are associated with T lymphocyte activation or autoantibody formation by the dysregulated immune system. Immune complexes together with the inflammatory cytokines cause the resulting tissue damage. Spectrum of autoimmune disease is very broad. It can be a limited disease like alopecia only restricted to the hairs or psoriasis only restricted to the skin, whereas it can be a multi-organ systemic disease like in lupus. It involves almost all organs. Multiple sclerosis, mainly it affects the CNS system, and so on in other conditions as mentioned here. What's the cause of inflammation and autoimmunity? So for autoimmune disease, there is a genetic predisposition. Like mentioned here, genes such as HLA-B27, HLA-C, IL-23 receptor can be susceptible for conditions called spondyloarthritis, psoriasis, psoriatic arthritis, rheumatoid arthritis, inflammatory bowel disease, and other genetic predisposition could be like HLA-DR4 for RA, HLA-DR B1 for lupus, and similarly, HLA-DR B115 can be a predisposition for multiple sclerosis. But many of us can have that gene, but we may not get that gene. It needs an environmental factor. Like a classical example is psoriasis, which gets after streptococcal infection of the throat. So the environmental factors could be infection, could be toxins, such as patients are getting lupus after exposure to hydralazine or some other drugs, and biomechanical forces, like many times we are getting inflammation at the site where the tendons are stressed, such as at the site of Achilles tendon. So under the influence of genetic and environmental factors, there is induction of inflammation, and this inflammation leads to condition like autoimmune disease. Disease spectrum, the state of the disease can be different at different time points, as it is showed here in this arrow. So you get, it's induced by environmental factor. At some point, there could be no inflammation. Then gradually, the disease becomes mild, moderate, and severe. So my main talk is that can we quantify inflammation? Can we determine the state of inflammation at these different points? And thus, can we make an early diagnosis, not only early diagnosis, how severe these conditions are? So can we quantify inflammation and determine the state of inflammation, different time of points? So these are some of the questions I raised, and similarly, it can come to your mind as well. Can we make a longitudinal study of an inflammatory process? Can we quantify the degree of inflammation with the progress of disease? Can we determine the real-time in vivo events, that exactly what activity is going on at the site of inflammation? Can we quantify the total inflammatory body, not only in one organ or tissue? Obviously, with current technology, it is difficult to do all this in human. However, can be done partially in animal models, such as shown here. We call it a collagen-induced arthritis. So here we are injecting to a mouse which is prone to arthritis with a different foreign protein. a chicken collagen is getting injected to the mouse, and that with time you are seeing with the day 0, the clinical CS is 0. You can see this paw is not inflamed. With time, the paw is getting swollen and red. Now, this is a clinical observation, so we can say this is mild, moderate, or severe. We cannot give an exact number, and we can do some quantification by doing a histopathological section, but then we have to sacrifice the mouse. So in one mouse, you cannot determine the degree of severity with current technology. So we raise the question that can we quantitate or quantify the inflammation by molecular imaging? So one molecular imaging is already exists, which is used for cancer detection. Probably all of you know it's called PET imaging, PET, positron emission tomography. Basically, the cancer cells are hypermetabolic. They use more glucose. So we inject radioactive glucose, 18F-FDG. Cancer cells being more metabolic, they will accumulate more this radioactive agent, and this radioactive signal can be determined. And basically it determines how much of the radioactive agent is there, how much millicurie of 18-FDG is there. It can be determined, and that's why we quantify the amount of the radioactive agent, and thereby we can have an assessment of the cancer spread. So what we did, we hypothesized that as inflammatory cells are also hypermetabolic, so PET imaging can be used to identify and quantify the degree of inflammation. And as you know, to save millions of people, many a times we have to kill tens of mouse. So we first determined to do this experiment in the collagen-induced arthritis model, CIA model. So this is the way we designed the study to quantify the progression and severity of mouse collagen-induced arthritis using an in vivo imaging tool, 18F-FDG, to do the PET-CT imaging and thus validate it against the standard histopathological evaluation, which I showed it to you. So this is here again the same picture. You are seeing that with time, the mouse is developing more arthritis, severe arthritis. The clinical score is going higher. And you can see here the inflammation between the bones is nothing. Here you're seeing minimal inflammation. And here you see the star with blue areas. This is showing diffuse inflammation. Similarly, you can see here the signal, the imaging signal. This red and yellow colors are getting brighter with the degree of inflammation. So this substantiate that this inflammation can be determined by PET imaging technique. And for this, you do not have to sacrifice the mouse at different points. This mouse PET imaging can be done on these mice again and again at different points. So once we substantiated that, we are doctors, so then we thought that we should be then able to not only determine inflammation, we should be able to see whether it is responding to treatment. So here you can see at the day 45 before treatment, and then after 10 days of treatment, day 56, you can see the inflammation is gone. And the mouse, which was not treated, you are seeing several degree of inflammation. Similarly, in the imaging, you see the imaging signal is going higher, 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 and following treatment, the imaging signal is reduced. So here we develop this imaging system, PET imaging system, a quantitative in vivo preclinical drug screening tool. And now this system we have developed in 5 years. Nobody is doing the conventional collagen-induced arthritis system, and most people are doing this model, and with time, the whole world will be doing this model. So this is to show graphically, you can see, so this is the PET imaging scores, day 28, the day 45, it is going higher, and then on the post-treatment, it is coming down, and it is correlating with the existing parameters. So we raised, can we make a longitudinal study of the inflammatory process without sacrificing the animal? Can we quantify the degree of inflammation with the progress of disease? Can we determine the real-time in vivo events? And can we quantify the total inflammatory burden? And here you can see we cracked the problem, and it is already getting used all over the world. So how do we apply now for the drug development? So many of you knew this molecule, cyclosporine, which is used for autoimmune diseases and in transplant medicine, and how it works, the T cell and the antigen presently getting activated by an phosphatase called calcineurin, and cyclosporine inhibits that, and that's how it inhibits the T cell. So I will not go into details of molecular mechanisms. So what happens, the antigen presenting cell and the T cell in autoimmune disease gets activated, as I said, and opens up the calcium channel, and then activates the calcineurin and activating the T cells. Now, to keep the calcium flow, the positive ions, you have to keep a balance. You have to take out some positive ions. They go out by the potassium channels. So Kv 1.3 and Kca 3.1. So as cyclosporine has a lot of toxicities, we thought that can we block the potassium channel to get a medicine like cyclosporine for autoimmune diseases, and we applied this technology. So there is a technology when you can when we can block or knock out a gene. So we knocked out the gene Kca 3.1, and then we tried to induce arthritis, same way which we showed. You see in the knockout mouse with our model, the elbows are not glowing up, the paws are not glowing up in the knockout mouse, but in the wild mouse, it is glowing up. So easy to show the efficacy and do drug development by using this model. So if this can happen in animal, why it cannot happen in human? So the similar model for arthritis, what we saw there are actually the model for autoimmune arthritis. Those models are autoimmune models for autoimmune diseases. So here you can see we have applied the similar model, the PET imaging model, and this is a hand of a rheumatoid arthritis where wrist gets more inflamed. You can see all these things are glowing up from the PET imaging. The signals are there showing the acute inflammation of the wrist. Whereas psoriatic arthritis, where the bulk of the disease is on the tendon, tenosynovitis. You can see this is the CT scan and this is the PET imaging. The flexor tendons, the extended tendons, all are inflamed with this technology. So this was experiments I'm showing in human just to substantiate, but we have to develop a technology to have a better resolution. When we did with the standard PET imaging machine, which is used for diagnosis of cancer, the resolution was so low. So we had to make a machine with a high resolution PET imaging focused on this joint. So after getting this success, we thought, hello?
Yes, we are hearing you.
I heard some sound. Okay, I can continue. Okay. So after finding that we can determine the inflammation, quantify it locally, then we developed this machine called the Exploral Total Body PET CT Scanner. This was made by the physics and the radiology department at our university to visualize and quantify molecular activity at small joints, periarticular, including the enthesis, and to determine the burden of arthritis in the whole body. So this is to give an example that Exploral Total Body PET Scanner clearly demonstrate the difference in disease pattern in rheumatoid arthritis, psoriatic arthritis, and osteoarthritis. You see, osteoarthritis is a non-inflammatory disease. It is only showing some degree in the big joints, whereas psoriatic arthritis is an autoimmune inflammatory disease. It is showing not only big joints, also in the small joints, and there is asymmetry. You see, these are the right and left hand. all the joints are not getting equally affected. Whereas in rheumatoid arthritis, we found that it affects symmetrically, it induces inflammation of the joints, and clinically we think the joints in the lower extremity does not get involved, but you can see that lower extremity also get involved, and the subclinical infection can be determined by this model. And you can see clearly here, this is a black and white picture, osteoarthritis, no inflammation. And in a patient with psoriatic arthritis, there is inflammation. And this is a measure, SUV max, to show the degree of inflammation. You see the SUV max is so high and in the patients with psoriatic arthritis. And so if we can do this, then we should be able to determine the response to the treatment. Here you are seeing a patient with a baseline with PET imaging, and you can see the degree of inflammation, the intense signal in the small joints of the hands and wrist, and after treatment, you cannot see it. So the degree of inflammation can be determined and quantified, and this will be helpful for drug development. And generally, you can see here, this improvement could be seen in 5 weeks, but clinically, the measures we do, that takes 3 months to do. So we think by this technology, we can quantify the inflammation much earlier, and not only that, this will help in various ways. This will help in various ways. This will reduce the cost reduction by reducing the duration of the drug trial, and also the duration of the drug trial will reduce. So there will be less exposure of these drugs and less toxicity. So again, that's what we do. We work on mouse, then we work on human, and then again, we go back to mouse if some more ideas come. So we found that the improvement could be shown in human in 5 weeks by this technology. How early this improvement comes. So we did another series of animal experiments, and this is the day 1, and this is the day 6. You see some of the animals are responding. They are showing response within days, within 6 days, whereas some animals are not responding. You see, their degree of inflammation is not changing. So this has been done so far, and we want to move on, and our future goals are mentioned in this slide so that this technology can be used widely. And as I mentioned, this will be a good model for drug development and preclinical discovery in the animal models, and as well, it will reduce the time exposure, the cost, and the side effects of the drug trials in human. That's our goal with this technology. And I want to stop here. Obviously, these projects were funded by the National Institute of Health, NIH, and National Psoriasis Foundation. And this is our group you can see here. And this is our PET CT and scan machine, which we have developed at UC Davis. Thank you very much again. And I shall be happy to answer if you have any questions.
Yes, thank you, Professor Raichaudhuri. Very nice presentation, and thank you for your continuous effort to improve the objectivity of looking into the autoimmune disorders using the PET CT machine. Here we have a question: what is the usual SUV of the small joints, and what is the SUV like when you are looking at the patient with rheumatoid arthritis present? This has been one of the questions.
So SUV max will depend whether you are doing in a focused area, and it will depend when you are doing in the whole body. So it does not have a fixed number. As I was showing in that animal model experiment, that it was varying between 0 to 6. The SUV max according to the degree of severity. So there is no fixed number. So
normally in a human being, what you take as the as your lowest SUV comparison, because normally we take for cancer like activity on heart, activity on other place as the baseline, and then look into other areas where the uptake is higher. So is there any such thing in case of human being having one of the arthritis that you take this area as your baseline and then look into it?
Yeah. So what we are doing, as you see, we have a comparison. We have you want to go back to the slide? You want me to put the slide again? That in osteoarthritis, I showed you the SUV.
Yeah, it was clearly black. Nothing was there. That's true.
Yeah, and also I showed you the graph.
Yeah.
In osteoarthritis, the SUV max was probably the whole body was less than 5 in osteoarthritis, and the median of that was around 15 in psoriatic arthritis. So, right. So you can consider that around 5 will be the baseline for any person.
So several salient points which you have made from your presentation with this experiment is that it could be a very good tool for developing drugs. And because the arthritis, the inflammation, the response to inflammation is picked up very early, about in the mice, about by 5 or 6 days you are seeing the response. Whereas in the other cases, it takes longer time if we want to assess it clinically. So you can say whether this drug is working, or you will cut the drug or put something else in case the inflammation is not going down. So that is on the management, obviously it helps. And third is the discrimination. That is, often we have problem when the arthritis begins, is that we know that psoriatic arthropathy is an asymmetric kind of arthritis in contrast to rheumatoid arthritis. But in the beginning, it may not be so clear-cut because, but here you have shown the tendons are the actually main areas where in psoriatic arthritis, the involvement happens in contrast to the joint. So in the pathogenesis also, your instrument will be quite helpful once it develops. One of the questions which remains is how often we can, normally it is very difficult to do repeatedly a PET scan on an arthritis patient. So we need something more advanced, means maybe more of your research, so that you have something like a PET scanner which is simpler and which can be done more often. Something like that. Plus your presentation itself, because today's meeting is on digital platform in healthcare. One of the important part of the healthcare, of course, is education. You have educated us through a digital platform already. So this is an exposition that medical education can be extensively done through this digital platform, whether it is immunology or any other subject, as we have been doing through webinars. If we have no other question, then thank you very much. And then thank you very much, Professor Raichaudhuri, for your excellent presentation.
Thank you, Dr. Ghosh. Thank you. And especially, you have summarized very well that the main use of this technology will be to develop drugs, but it can be used to diagnose a disease in an atypical situation. Like, many times a patient can have, let us say, psoriatic arthritis with psoriasis, whether the patient has a psoriasis or not. Just it could be osteoarthritis and psoriasis. In those situations, it can be used as a diagnostic tool. Also, you have clearly, mentioned these points. Thank you for comments.
Just before the next speaker starts, Dr. Ghosh, and
yes, please.
Just one quick thought. As you talked, I was thinking, although I'm a cardiologist, so far the rheumatologist, I'm absolutely sort of a stupid fellow, but like a stupid person asking a question. So please excuse me for the question, but as I was thinking that you are trying to compare, let us say, degenerative arthritis versus, say, inflammatory arthritis, and say we have taken rheumatoid arthritis, then atypical asymmetrical arthritis like psoriatic arthritis, which is probably some kind of immunologically mediated arthritis. Now, we also know that there are many arthritis which is something rheumatoid look-alike. Even I am quite aware of such a single joint arthritis which eventually turned out to be rheumatoid arthritis. There are case reports. Now, there are situations when even the rheumatoid arthritis may look different. The point is that when you try to capture the inflammation on inflammatory sort of images by PET scan, is there any kind of gradation by which you can actually say that, well, these are the images which is extremely characteristic of rheumatoid? These are the kind of little variation of those images which may not be exactly rheumatoid, but could be rheumatoid lookalike?
Yes, you're right. And that's one of the purpose, am I right? That here you can quantify the degree of inflammation. So if you see, and if your SUV max is high compared to the baseline, that first would say that this is an inflammatory disease. And then the rheumatoid arthritis, as Dr. Ghosh mentioned, causes more of a synovitis. So if we see the synovial inflammation, not much enthesitis, then probably we can say that, yeah, this is a very atypical form of rheumatoid arthritis. So in atypical condition, we do not need this PET imaging to diagnose a rheumatoid arthritis. But in such situation, you are explaining a monoarthritis. Many a times in rheumatoid arthritis, it can present as a monoarthritis. So this technology can be helpful for that too.
Right, Professor Raichoudhury. Thank you very much.
So you may invite the next speaker, Dr. Amit Dinda.
Yes, Robin, Amit is your class friend, so introduce him.
Sure. So
he's still in AIMS, or he has retired and gone somewhere.
Yeah. So it's a great. I think he's a sort of the man of nanotechnology in India, probably South Asia region. He's a hero, and it's a great pleasure to invite Dr. Professor Amit Dinda in this important meeting of Bengal Chamber of Commerce, and he's going to talk to us and educate us on emerging role of nanotechnology-based innovation in healthcare. Dr. Dinda, please.
Thank you. Thank you, Dr. Chakraborty. And really, I'm grateful for giving me the opportunity. So today, what I'll do, because we work in very large spectrum of the nanotechnology application. So just in 15 minutes, I will give you certain examples from our lab, the technology which we have developed, and particularly 3 of it are going for the clinical trial, and there are several of them in the pipeline. So this will actually explain that this technology is basically enabling technology. You can apply for any kind of the health, the care system if you want. So So the concept of nanoscale, I think all of you know. What we do, we create the mass material into a nanoscale, which is around a size of a virus, 100 nanometer. Sometimes we use 10 to 20 nanometer, and sometimes we use also a little larger one, depending upon the type of use we want to do. And we choose the material and functionalize in such a way that it can do very specific function inside the body, outside the body. And if you really look into the nanotechnology application, now I can't tell you, you cannot, there is not any area where nanotechnology can be applied. It can be used for making aeroplane, the advanced composite, to vaccine development, particularly for COVID. There are solid lipid nanoparticle based, several vaccines are very successful in the market now, in the use now. So with these, see, if you look into the mass application of the nanotechnology, see what we are moving into, we are moving from these large, the technology and instrumentation, we are miniaturizing it into a nano form. And so the whole spectrum of this technology is moving towards nano. And if you look into the healthcare is one of the area, but in electronics and in the material science, in energy, in all other areas, even the textile also, the nanotechnology has done a quite great job now. If you look into the investment, particularly as it is the forum of the Bengal Chambers of Commerce, if you see in the investment wise in 2020, because of COVID, it may be a little less, but almost $3 trillion has been invested in this area of nanotechnology. So far as the health is concerned, the application of nanotechnology can be very diverse. We can use it for imaging. We can use it for the different kind of targeted therapy. We can use it for the point of care devices, the nanobio chip system, the nano sensor, which we can embed inside the body, integrated with the IoT devices. So there are a huge number of applications. And when you amalgamate together, we are nowadays telling them that it's emerging technologies in medicine. Now, if you look into the emerging technologies in medicine, the major problem we face nowadays is that how do we assess their safety and efficacy vis-a-vis quality? That means regulatory approval. Who will give this approval, regulatory approval of this? But then in any case, things are moving very fast. I'll give you example. Large number of these nanotechnology-based the systems and drugs are now is being used in the market. Now, in AIMS, I think what we did over the years, we have developed a very, very interdisciplinary area of nanotechnology research. We have picked up areas of diagnosis in the area of imaging, molecular detection system development, nanobiosensors. Today, I will not give you all the examples because of lack of time, and also drug delivery systems, which we have developed now. So this is going for clinical trial. Now, we have developed the large array from the magnetic, the iron nanosystem, that is SPION, supraparamagnetic iron nanoparticle. And in fact, we are using this system for clinical trial. This will start very soon, is for the spinal cord injury repair in human. I will not be showing that data today. But then the hormonal system and many other systems we are now developing are being used for different kind of technology. So this is the array of things we are working now. And but at the same time, as I told you, to any of this technology from the lab, it has to be the bedside, then we have to also go for regulatory science research, so nanotoxicity studies. So that area also we are working quite actively. Now, give you very few examples. Now, one of the most successful things which we do is the oral amphotericin B delivery system. Now, here we have used a cholesterol chitosan conjugated nanoparticle, which has got a very high the oral absorption capability. We have shown its biological equivalence, the pharmacological equivalence, and therapeutic equivalence in comparison to the ambisome, that is the widely used liposomal preparation of the amphotericin B. And it is much less toxic than the ambisome, and its oral bypass systems and its tracing also we have shown, and that is obviously it's now going for clinical trial. Apart from that system, we have got the several macrophage targeted delivery system which can be used for the viral infection as well as tuberculosis. But obviously this I am not pushing through because it is much more complex. The different nanogel carrier system we have developed. One of them we are using now in the dual phase hybrid system. So what we are doing, we have picked up one the silk fibroin based the gel, which has got a sol gel kind of the mechanism. It is outside the body. It can be a gel kind of thing, like a shampoo like consistency, where we are dispersing or mixing the gelatin nanoparticle containing your anti-cancer drug, and then this can be injected by ultrasound guidance inside this tumor, and then the drug which will be the coming out of this nanoparticle gradually will be dissipating through these the other the gel system. And the advantage of that system is that not only it can spread in the localized tumor area as a local drug delivery system, but also it can flow through the lymphatics and kill the metastatic cells in this area. But anyway, so there are several things. Because of time, I'm not showing you. The other interesting thing for tuberculosis, when we are requested to develop a nanosystem, the major problem which we realize is that the people are trying to develop anti-tubercular the delivery system, but then they are mixing the different drugs with different nanoparticle and mixing them together and giving it. The problem is that you cannot create or predict that the particle carrying INH and particle carrying rifampicin will go in that same ratio of biological availability which is needed at the area of the disease. So that's why the technology which we have developed is that 4 anti-tubercular drugs, any of the drugs, in fact 4 drugs, you can develop, you can incorporate in one nanoparticle by layer technology, and we have patented it. I think the things are going on. The cancer nanomedicine is the area where maximum amount of these drugs are nowadays in the market, and this is the typical success of these nanomedicine. And I think all of you know there are the different preparations of doxorubicin, paclitaxel. I'm not going to the details. Some of these drugs which are approved and in the clinical trial, there is a whole list of it, and more of them are going to come in near future. So our lab also developing. That's why the different the hybrid system of the cancer drug delivery system. In fact, the latest one which we are developing is that the ultrasound sensitive the drug releasing system, where we are putting our drug into a polymeric system, and inside that nanoparticle we are putting some of these material. When you put ultrasound, they expand. So from the liquid, it turns into a gas, and then they burst the polymeric bubble, and so that it can induce the drug release. So that means the nanotechnology applications you can really engineer in a different way. But the major constraint in that is that you have to use the systems which are US FDA approved. In the sense, all the material, your impurities, everything has to be controlled. So there is a big challenge for that, but it is possible to do these things. So, what is the context of India? You know, in India, there are several companies who have already developing the nanotechnology-based the therapeutics. But in this, I think I wanted to show that also it is progressing quite well, and many of these industries are now trying to invest money in the nano area. I told you in the beginning, the major constraint is that our regulatory approval. Till in India, unless, unlike US FDA and European Union, the nanotechnology-based system is very difficult to pass through the regulatory agency. So I'll give you a few of the examples in our lab. So we worked with the gold nanoparticle and developed the particle for imaging. One of the major problems in the gold nanoparticle is that if you make the size little larger, more than 10 nanometer, then the chance of sequestration inside the body is quite high. And so we have shown that using nanoparticle which is less than 10 nanometer and less, it actually goes out of the body through urine very quickly. And in fact, it is possible to do the image intensification in our mouse animal model tumor. But the problem is that we are not pushing this technology because the need of the amount of gold nanoparticle for a good imaging in human system will be pretty high. So though we have developed the technology, we are not pushing it. Similarly, we have developed another technology where as a contrast agent for MRI, we are using the iron oxide nanoparticle. This is already done by some of the companies like DuPont, but then our system is very different. We have a targeted imaging kind of system, and we have published it, and the more work is going on in the system. Now, for the in vivo near infrared fluorescence imaging, I think a little different type of imaging system, which Dr. Raichoudhury has shown. So there we are using some of these, the fluorescent molecule, which has got travel. The fluorescence can travel more than a 12 centimeter kind of situation. And these are usually near infrared based imaging system. And we have used quantum dot, but then quantum dot cannot be used inside the body. So we use these polymeric nanoparticle doped with some of these the near infrared imaging. In mouse, it is working very well, but I think we have to go a little far off for using for human imaging, and we have. I think in the future it will be possible to do. This is a BIDAC project we are working with, where we have done what the cytokeratin conjugated the nanoparticle we have tagged with technetium-99. And then for sentinel lymph node detection, we are hoping that it will work very well. So in that case, we can use the sentinel lymph accuracy will be much higher, and we can using technetium-99, the sensitivity and specificity is also is increased quite a bit. I think it is clinical trial has to start, and then we can tell you maybe within 6 months or so. So I'm not going to the detail of other work of our molecular imaging as well as theranostic, but the vaccine development is one area which we worked, I think 2 years back. And what we did, we have used the polycaprolactone nanoparticle of around 14 kD, and these are around 60 to 100 nanometer in diameter. We have loaded the antigen. We worked with hepatitis B vaccine. The advantage is that this is a system which goes through orally and disseminate in your all lymphatic system. So its antigen presentation very robust, and with a single shot, you can go on stimulating the antigen in mouse system. We have shown mouse and guinea pig almost 6 months. That means you don't need any booster dose for this. And this oral vaccine platform is also, I think we are getting the permission for clinical trial. This system actually, very recently we are initiated the work to try with the Covaxin. I mean, sorry, not Covaxin, the COVID-19 antigen. And we see that how also whether it can work or not, because antigen presentation capability of this oral vaccine is pretty high. And in this figure, you can see that it goes through to the intestinal villi, to the lymphatic targeted. It can go through these interstitial lymphatics and also go to the inguinal lymph node, mesenteric lymph node, and other lymphoid organ. And we have shown that it can stimulate there for 6 months or so. So quickly to another area which is also quite interesting is your photothermal therapy using nanoparticle. We have published long back that gold nanorod is very useful for localized photothermal therapy, and we have proven that in superficial cancer in the skin can be treated with that. But the problem is that it is highly toxic, so it cannot be used inside the body. So another technology which we developed is that superficial bladder cancer is very difficult to treat that way. So what we did, we have developed this gold nanorod, and they cover it with the alginate, which is the mucoadhesive polymer, and load mitomycin C in those polymer. So if we just raise the temperature around 42 to 43 degrees Celsius, these gold nanorod, because of SPR effect, it produces enough localized heat and that release mitomycin C locally and kill these the superficial bladder cancer cells, which sloughs off in the urine. So this is a totally in vitro kind of application of the technology. We're using gold nanorod, but it is not injected inside the body. I think similarly, there are several work with the gold nano system. I'm not going to the detail. Then also radiopharmaceutical area has got a huge scope with the application of nanotechnology. And so apart from our lab work, I think a couple of things I want to present, which is also have a huge advantage, is that oligonucleotide drug delivery through nanosystem. We develop a system of oral gene delivery, but this is another area where USFDA has already cleared several of these nanosystem-based oligonucleotide delivery, but they are not for systemic use, mostly for the eye. Some of these things are under clinical trial for spinal cord injury and so. The major challenge which is coming up to us is that as we are developing very advanced nanosystems, so they doesn't fall in the typical of the pharmaceutical ingredient which you're using in the form of our current practice. So they become more complex. So basically, so nowadays we're using the term of non-biological complex drugs, because sometimes we're using polymer, metal complex. So how to actually push through them is a big challenge. And if you see, there are a lot of these regulatory agencies are already initiated the talk about that, and because these are really, really helpful, and not all of them are very bad. Many of them are pretty safe. They have got a safer window also. So for India also, we have developed these guideline, and I'm one the active person in the developing this guideline, which is taken up by the drug regulator DCGI, CDSCO, and we hope that very near future the Indian regulators also will be able to clear our nanotechnology-based drug and other technology development. So I feel at the end, this is my last slide. So I feel that this is a technology which is a very, very powerful technology. But the problem is that when we develop the technology, when we go through the animal trial and then coming to the clinical trial, see the data collection when we do, we know that there are certain kind of innovation which is definitely risky. So I think we should not move them. So they can put in the red basket. And there are certain innovations which are pretty safe. So they should be put in the green basket. And where we are not sure, the scientists are debating among themselves whether it is risky, non-risky, then uncertainty is there, then we can stop them in the yellow basket. So in that scenario, there are several kind of application for healthcare. I think I have not presented our work on the human elastin-like protein-based system, which is a extracellular matrix protein in the nanosystem. So there are a lot of things can be done also in the regenerative medicine, nano-regenerative medicine interface. So the conclusion is that it's a huge potential of application of this technology. Thank you very much. I have got large number of the the funding agencies. So all of them, I am grateful to them. Thank you for your attention.
Thank you, Professor Dinda, for a very excellent talk. And as you have rightly said, the nanotechnology is cutting across the technologies in not only in medicine but also on every sphere of life. And see, the oldest nanotechnology platform, which we never talked about nanotechnology, was the colloidal gold car.
Yeah.
So that time we didn't say nano, though it was nano. And using the same kind of technology now, as you rightly said, there has been explosion of near patient testing. That is, the doctor can test pregnancy at the door. We can look for sickle cell anemia at the door. Many infections and infectious diseases can be tested. Plus, therapeutically, you have showed a huge advance which has been made using nanotechnology, particularly in reducing the toxicity of many of the chemotherapeutics which are used in cancer and increasing its total action on the cancer cell. Means after they go out of the blood vessel, they stay there for a long period of time. So it is a mind-boggling work you have been doing for us in India. Atmanirbhar Bharat, you are doing so. It is a great thing, and I congratulate you. Robin asked one thing. I started doing one thing almost 10 years back in the area of cardiology, where I labeled spion, that is paramagnetic iron particle with 7E3, that is an antiplatelet antibody, in order to image the image through MRI the various thrombosis. so platelet gets tagged by the spion, and wherever thrombosis happens, platelet go. So this is one of the area I could not develop because in my place I didn't have enough wherewithal to do it. So I did it in animals, and this is the area in which you can image thrombosis in various areas of the body, and of course in various parts of the heart.
Yeah, exactly. So basically, I didn't show it's a difficult work. In fact, some of our failure. Let me tell you. So, but after we mastered the macrophage targeting, I developed a siRNA based delivery system, and for the initial plaque formation, macrophages can be targeted through that. And then we have shown that it in fact arrests the atherosclerosis plaque progression. In fact, it reduces it. The problem came when we, after rat and mouse model, when we tried to go for higher animal, then we realized it's not so easy thing to deliver.
Yeah, that's true.
So, but then I agree with you that there is a huge scope of the cardiovascular disease also, both for imaging and also the diagnostic front, and as well as in the therapeutic front, the nanotechnology application can be developed. And so it depends upon that what is our very focused area where. So normally what our how do we go about it is that we try to see that what are your solution currently.
No, you are doing work on almost very many areas. So in this small amount of time that you have to present, you have presented, you have packed the data of areas beginning from cancer, beginning from regenerative diseases, diagnostics, therapeutics, imaging. It is really, as Robin said, is a mind-boggling work you are doing. I congratulate you. Keep on doing this. And as I said, help us in the Atmanirbhar Bharat program as our Prime Minister is looking for it. And obviously, nanotechnology will find its use in many places. We have not actually, we probably will have occasion to talk. So, Robin, we have any other speaker?
Yeah.
So before you go to the other speaker, I have a question for Amit. I know Amit for many years because we are from the same college and we work together at AIMS and PGI. So and also we participated in some project in nanoparticle with Amida. So one thing which Professor Ghosh has already mentioned, that this nanotechnology will allow the drugs which we are using currently in much lower dose, and that will reduce the side effects. So Amida, we were working on methotrexate delivery for rheumatoid arthritis. And as you know, methotrexate is not only used for rheumatoid arthritis, it is widely used, cancer, autoimmune diseases, diseases of the old age. How much did you progress? Because, we did that project partially together, and it showed a very good result. Did you make a patent? Is it coming to the market?
Yeah, so basically, I didn't show that data today. So basically, we have developed this system pretty well. In fact, we have filed in the process of filing this patent now, and the advantage is that this has got a very specific the high dose targeting of these inflammatory macrophages. What we have done. And I think we are still, I think, couple of months we need to work on that, because a little bit of off-target effect we have to minimize. But there is a huge scope in that. So obviously I'll get back to you, because the PhD the student has completed, but I think next phase of work I'm planning to start. And in that, I just want to quickly tell that I'm lucky to get a bioincubator to be set up at All India Institute of Medical Science in near future. And then I think we'll get a great chance to work with the industry interface at All India Institute of
Keep doing that, Amit. Congratulations.
Thank you. It's a wonderful presentation. I think we are the last speaker of this session, as well as the last speaker of Health Tech 2021, Professor Sriti Kanna Raichaudhuri. And may I request you to present your talk on this Health Tech meeting, Dr. Sriti Kanna Raichaudhuri.
Hello.
Yes.
Please. Yes, you are audible.
Okay. So I'll talk on molecular technology, bench to bedside, as all my previous speakers have focused on. I'll be talking mainly on bench to bedside, and with an aim to development of novel therapies for inflammatory diseases. So here is, I'll give a brief description of how the immune-mediated inflammation occurs in autoimmune diseases. The memory T cells, this is a memory in relation to a specific antigen. These are, developed in the primary lymphoid organ, and then through the blood vessels, they go to the specific target organs, like to skin for psoriasis, to bowel for inflammatory bowel disease, joints for arthritis or spondylitis. And the process of going through the blood vessels to a specific organ is a process called homing mechanism, and this happens through a specific interactions between the blood cells and the endothelial cells. And so here it's in the cartoon, I can show you that like the blood cells go through the endothelial cells by adhesion and go to, in the specific tissues, there are chemotactic factors, and they do a chemotaxis of these cells. And then they go to the tissue, and the specific antigen-presenting cells, they attach to these lymphocytes through through TCR, and they also need another stimulation through CD28 antigen on T cells, and then they release a lot of cytokines, different cytokines, chemokines, to make the inflammatory process. So here are some examples of different inflammatory diseases which we are working on. This is psoriasis, and there is a very specific phenomenon called Kevner phenomenon, where at the site of injury they develop this disease, and this person had this is his wristwatch area, and the wristwatch was a stainless steel watch, and we did. The patch test was done, and it was negative, and it was a psoriasis, and this is a very specific phenomenon for psoriasis. This is systemic lupus erythematosus, and this is a rheumatoid arthritis synovial inflammation. And here in the ankylosing spondylitis, you can see the inflammation in the spine. So the paradigm we are working is like the disease process is the cells are adhering and then migrating through to the specific tissues and getting activated and releasing cytokines and causing proliferation, inflammation, and tissue destruction. So we wanted to see whether we can see this as a proof of principle study. And what we did is we used that Kibner phenomenon technology, phenomenon to establish a psoriasis in a psoriatic patient by doing a Scotch tape abrasion. And here, and then we did serial biopsies, first week, second week, third week, fourth week, and then a chronic plaque developed. So here in first week, you can see that there are cells around the blood vessels. And in second week, you can see there are more cells coming. And in third week, they are kind of invading the epidermis. And when they are going to the epidermis and there is the site of activities and the keratinocytes are proliferating and it started protruding, and this depth of the skin is increasing. And here you can see the activities are much more robust, and after a few weeks it becomes a chronic plaque, and with this typical psoriatic histopathology. And so if this is what we found out, then how do we stop this process? So that is where we are working to develop a different system to stop this process. So, like as we said, that the T cells plays a very important role in this inflammatory process. So how do we know that like these T cells are going to different tissues and whether they're unique and they have unique characteristics? How do we characterize these living cells and find out their functions? So here is Dr. Len Hartzenberg, who developed this technology called fluorescent activated cell sorters, which is FACS, in short, FACS. And he developed this in 1960s. And as you see, the professor is young and the instrument is young too. And it's like a very big instrument. He's covering almost the whole half of a big room. But with time, this technology has improved, and it's now a small instrument. You can put it on a bench top and use it in the field also. So we worked with Dr. Hartzenberg. We were very fortunate to work with him for almost 10 years. Oh, what is happening? So here, I wanted to show you the power of this high-dimensional FACS technology, that we can take the blood cells and then find out a living cell from there. I'm not going through the details of this process, but you can find out the living cells, and then you can characterize them using different molecular stains. And then find out the characteristics of these cells. And in inflammation, the cells are activated. So we wanted to see whether they can they express these activation markers, which is HLA-DR, one of the activation marker. And with all these process, we could find out that like these cells are CD3 positive cells and CD4 positive cells. They have HLA-DR marker, which is the activation marker, and they are memory T cells. These are CD45RA and CD11A cells. And in this plot, we are talking about this TRK, which is a nerve growth factor receptor, and we are in the process of developing some treatment focusing on that area. So here I'm showing. So we found out that characteristics of these cells by the phenotypes, and we wanted to see how they work, whether they're functional or not. And so that for that, these live cells are very important. And here we are showing that like IL-17 expression, which is very important cytokine in the process of inflammation cascade. And we showed here that like these CD4 positive cells, which are memory T cells, and they produce IL-17. So here I just wanted to show you that these IL-17 producing cells, but they also need another stimulus to become as Th17 cells, which produces IL-17. And then they attach to they go to the target tissues and attach to them with this IL-17 receptor, and they produce signals, and different tissues react to that signal differently, like keratinocytes. They produce cytokines, they have inflammation and proliferation and causes psoriatic plaque. And synovial fibroblasts, they cause the proliferation of the synovial cells and produce different cytokines, causes bone destruction, and leading to arthritis. And then endothelial cells. So whenever there is a proliferation, we need food so there is angiogenesis. So there is proliferation of the endothelial cells. And they also activate these osteoclasts, which causes bone erosion. So this is a summary slide for how this IL-23 and IL-17 cytokine axis work in autoimmune diseases. So, after we find out the characteristics of these cells, we have our main aim is to develop treatment for these diseases. So, we developed this animal model for psoriasis, with the focus on this psoriasis. But this is a skid mouse, which is deficient for T cells and B cells. And so they accept graft quite easily. And what we did is we took a biopsy from the psoriatic lesion from human, and then we transplanted on mouse. And after 3 to 4 weeks, it get accepted, but it still maintains the features of psoriasis. You can see the scalings and redness. And if we do histopathology, it shows the typical features of psoriasis. And this is an H&E stain. You can see the ICAM, the adhesion molecule, these activation markers on the T cells. And this is the nerve growth factor. We wanted to show you this. That was the work we were. So it maintains all these features of psoriasis, and it maintains up to at least 6 months, which is sufficient time to do tests for drug development or efficacy of a drug in preclinical setting. But this is a very complex because it involves both human and mouse. And as you know, that psoriasis is a human disease. Other diseases we can replicate in mouse, but in animal models. But it's very difficult to replicate that in mouse for the psoriasis. So this is a model. It's kind of humanized mouse now. So here, I just wanted to reiterate that like these T cells and antigen presenting cells, their activation works more for full activities. They need two signals. One is this TCR, the T cell receptor, and then the second stimulation is with the co-stimulation with CD28 molecule. And here is with just a cartoon to show you that like these two American brothers ran with bulls in Spain, and then later they cherished their life and celebration and brotherly bond. And so like these two horns of, just to describe the two horns of these T cells that needs to have a full activity and full effect of it. So, once we developed this animal model and we wanted to, our main aim is to develop and show whether these, all these activities, we characterize the T cells so that really works in a, we can show as a proof of concept whether this really works in the drug development process and it can be used as a treatment. So we worked with this anti-CD28 antibody, which is an humanized Fc-silent antibody, and we treated this this kid mouse with this drug FR2-55734 for 4 weeks, and then this is before treatment biopsy, and this is, you can see how it improved after the 4 weeks of treatment. So this drug was in developing process. So we also worked on this IL-23, IL-17 pathway to see and developed several drugs, and we have contributed significantly with this drug development of different anti-cytokine antibody. And they are used in psoriasis, psoriatic arthritis, rheumatoid arthritis. So here, this slide I showed you before, that how different steps we can make an antibody or a treatment compound to stop that process. So here we can, this is an IL-12 and this is IL-23. And you can use developed antibodies against these IL-23, which is specific for the IL-23, this P19 portion of it. And this guselkumab, tildrakizumab, risankizumab, these are the antibodies which can be blocking the IL-20. You can stop the IL-23, that very upstream process. Or this ustekinumab, this actually stops with this P40, which is common for both IL-12 and IL-23. And this secukinumab, ixekizumab, these are inhibiting the IL-17. And this IL-17 receptor inhibitor is brodalumab. And when these T cells and then attached to this target tissue, they do a signal transduction, and that's how all this process starts. And this process can also be stopped. And right now there are tofacitinib and upadacitinib. They stop the use the JAK-STAT pathway, and they stop that process. Angiogenesis is stopped by through RR gamma T cells, and there are many other mTOR inhibitors. So I just wanted to summarize that, like, this is what it takes, that like you find out the mechanism and try to develop something to stop that process at a very specific time point or a very specific step to stop the disease process. and molecular technologies and are very much advanced nowadays, and we can use those technologies to develop drugs and go through the process of a safety test in animal model, and then a phase 1, phase 2 study to find out the safety and efficacy of the drug, and then for a bigger clinical trial for human use in clinics. And molecular technologies now is a great help to do those processes. So I'll stop here. Thank you very much.
Thank you very much, Professor Mrs. Roy Choudhury. You have illuminated a very complex area of both understanding the pathobiology and from that understanding developing drugs using a combination of facts and an animal model. And I wish you all the success for subsequent final trials in medicine, because we are looking forward to it. And I now invite, if there is any question from the audience or from our faculties, other faculties, if you would you like to ask any question to her?
Any comments from the panelists? Because that was a very, very elaborative talk. But for me, it was really complex, to be very honest with you, because I do cardiology and but Professor Rai Choudhury, your original work is phenomenal and very, very, very impressive. So that's what I like. And I'm sure that, with your contribution as a professor of infectious disease and medical microbiology, as you had your training from All India Institute of Medical Science, followed by United States position, and especially working at both California North State Medical College as well as University of California, Davis as a research worker. But to be very honest, for me, it is really, really complex. If some of you can make it simple for me, so that would be really great.
This is Robin, what she is telling. Robin, hello, Robin.
Yes.
What Mrs. Raichoudhury, what Mrs. Raichoudhury is telling, that in case of your autoimmune myocarditis, right, she can isolate by fax the T cells, look into the circuit, and develop medicines. Using that kind of platform, you can look at any autoimmune. She has described on psoriasis and skin patches, but this can be done for any other diseases using a combination of flow sorter, sorting the culprit T cells, seeing what they produce, using them on a skid mice as an animal model to see what kind of therapies will be effective. And finally, you take that therapy directly to phase 1, phase 2, and phase 3 clinical trials.
Excellent. In fact, Professor...
Excellent. Thank you.
Professor, actually, this one slide was very, very impressive when she talked about the T cell mechanism in the kind of inflammation. I was just thinking about the COVID. Is it possible for you to explain that post-COVID halt, many patients after COVID, they are getting some features like arthralgia, generalized aches and pains. Has it got anything to do with the T cell?
Well, that's exactly what we are studying, right now. And as you know, it's a multi-system disorder, and these inflammatory cytokines, there is a huge surge of these cytokines, and that causes the multi-system disorders. And especially at the beginning, it was found out in children, even though the children get very few infections, but the rate of these multi-system disorders were much more in the affected children. And then we found out that this prolongs the disease, the after effect of the disease in adult population. And this happens through all these array of cytokines developed, and it just stays on. So that's what we're working on to find out whether we can, develop something to stop that process.
Oh, excellent. Dr. Dinda, do you think that your nanotechnology can intervene in such complex cytokine disaster in human body?
Yeah, I was listening to Shriti's presentation also. So the one interesting component I think I want to mention, I didn't elaborate. So if you think about precision medicine, so there are now a couple of components in that. What Dr. Shriti has pointed out, that is the molecular target. Exactly, we're identifying where is our target should be, and then we are targeting that. Now that is a target biomarker we are identifying, and we are trying to locate and address that. Now the carrier part, where your precision, the targeting can be aided by nanotechnology. I'll give you example, that's oligonucleotide based therapy, where we are using the nanovector to definitely homing it as those sites where it is needed. I'll give you example, certain situation like there are certain the irreversible liver diseases, even maybe I have not the reviewed, but even maybe cardiac diseases, it may be a little complex. But in liver diseases, people are trying to actually target through the nanoparticle the specific receptors which are expressed highly in the hepatocytes in abnormal situation. Say, for example, in certain kind of hepatitis. And then try to actually reach the molecular level inside the hepatocyte to alter its metabolism or whatever the defect is. So those kind of things, obviously nanotechnology is helping one step ahead of same molecular targeting it is doing, but helping the different biologics or different molecule to reach that specific site. Excellent. So obviously combination will be very, very useful.
That sounds really very promising. Taking it further, would you reckon that the CRISPR technology, if that can be brought in to interfere this T-cell and cytokine complexity, do you think the CRISPR technology may have some role for a permanent solution?
Well, that technology is very, very useful technology and advanced technology, and this has been used for, gene editing. So hopefully we should be able to use that to stop this whole process at the beginning.
Thank you. Dr. Ghosh, I think we are probably finishing the session. Would you like to
Yes, we had a great time. We had a great time, and we listened three very different, distinct areas on immunology as it cut across the healthcare and medicine. And I would like to thank all the speakers for their excellent talk, the Bengal Chamber of Commerce for inviting me to be with you at this very important meeting. And of course, my friend, Dr. Robin Chakraborty, for getting me into it. And I thank you, Robin, very much for all the good time I had in this evening. Thank you very much.
Thank you, sort of all the speakers, Professor Shiva Prasad Rai Chaudhary, Professor Amit Dinda, and Professor Sritikona Rai Chaudhary for a phenomenal presentation of molecular imaging, nanotechnology, and molecular technology, and very basics. And that we have to address that probably in the technology meeting, in health tech meeting. Such kind of information are extremely useful. But it is also important to recognize that these technology has to be taken into clinical application so that eventually the patients can be benefited, wellness can be improved, and human life can be much more better from many diseases. With that, I thank everyone, Professor Kanjaksha Ghosh, Professor Shiva Prasad Rai Chaudhary, Professor Amit Dinda, and Professor Sritikona Rai Chaudhary. So we had a great session, and I therefore would like to close the session and request Angana Guha Rai Chaudhary to take over and conclude I conclude the meeting, and she can probably take it further and conclude the Health Tech 2021, the 4th Health Tech conference. Thank you, Professor Ghosh, and thank you all the panelists. Let Angana come and conclude the conference. Angana,
thank you, doctors, and thank you, audience, who are with us still till the end. And this brings us to the conclusion of the 4th edition of Health Tech. We look forward to having your participation in our forthcoming programs. Stay in touch with us, follow us in our social media, and we'll also be in touch with us for our various initiatives in the related areas. Thank you. Closing the 4th edition of the Health Tech today. Thank you.
Thank you, ma'am.
Thank you.
Thank you so much. Thank you.
Thank you.